Good evening, everyone. My name is George O'Toole. I'm a professor of microbiology and immunology at Dartmouth Medical School and the co-director of the microbial diversity course here at the MBL. It's my pleasure and my privilege to introduce my colleague and my friend, Dianne Newman. I think it's typical to start these sort of introductions listing the kind of accolades that the speaker has won, and Dianne's won many of those. The Eli Lilly Young Investigator Award. She's a fellow of the American Academy of Microbiology. She was an investigator for the Howard Hughes Medical Institute and a fellow of the MacArthur Foundation and a named professor at Caltech. She's had a long history with the MBL and served as a student, a TA, instructor, and co-director of the microbial diversity course. But I don't think that list really captures who she is. If you ask her students, they'll tell you about her immense enthusiasm for microbiology, and you see it in one-on-one interactions and when she gives lectures, and you're going to see it tonight. If you ask the many postdocs that have worked in her lab that have gone on to faculty positions, they'll talk about the amount of work and intense support she gives to them when they're in the lab and when she leaves the lab, and her support for young scientists in general. If you ask her colleagues to tell you about her work, they'll tell you about the insightful and often synthetic work that she's done at the interface of many fields. For example, Dianne started as a geochemist and came to work with Roberto Coulter, which is where I first met her at Harvard Medical School over 20 years ago. And she really developed a passion for molecular genetics and trying to understand genetic processes. And I think unusual is the right word when someone wants to fuse the study of rocks and the study of microbes and can do it successfully, and she has, and really launched new fields in doing so. In recent work, she's done it again, combining the work of geochemistry and geoscience and applying it to the medical field, studying chronic infections, cystic fibrosis. And I think you'll maybe hear a little bit about that tonight. So in tonight's talk, Dianne's going to talk about life without oxygen. And for the multicellular eukaryotes in the audience, and those of you who study multicellular eukaryotes, you don't typically think much about life without oxygen. And that's probably because we don't do so well when oxygen is missing. But for the vast majority of diversity on this planet, and if we can sum it up this way, all the stuff you can see that's alive could be represented by this little circle, and all the microbes by this big thing right here, whatever you want to call it. It's my big V here. So the vast majority of microbes on life, or life on the planet, is microbial, and many of those microbes have a perfectly content time living in the absence of oxygen. In fact, some of them are killed if there's oxygen around. So what Dianne's going to do tonight is take you into a world that you don't think about very much, that you can't see, but has an immense impact on the entire biosphere of our planet. So now I'm standing between you and Dianne, so I'm going to finish up by saying that Dianne has a long association with the MBL, a long and warm association, and has an immense respect for the educational mission, the world-class educational mission in science that's done here at the MBL. So I know she's a little nervous before she gets up and gives her talk tonight, but I think that's going to be okay. I cannot say how proud I am and how excited I am to see her talk. And you guys are in for a treat.
Good evening, everyone. This truly is a special night for me. As George said, I've been around the MBL long enough to know what a distinguished group of people have given this lecture, and I must say it's both an incredible honor and somewhat surreal to find myself now on this side, looking out as opposed to in the audience. So thank you very much for inviting me here, and it is a wonderful way to begin after a friend introduces you. Not only a friend who knows me well and for whom I have a great deal of academic and personal respect, but also someone with whom I share a total passion for microbes. And so George basically gave you the punchline of my talk, so we can fast forward now through the first few slides. I'm not going to do that. In fact, I think microbiology, and particularly the wonder of microbial metabolism, is worth celebrating as much as possible. So I thank you for that great introduction, and now I will get into the meat. So the title is, of course, Life Without Oxygen. And I thought I would introduce you to the remarkable ways microbes have evolved to make a living in the absence of oxygen by first reminding all of you how dependent we are on oxygen and why. And a dramatic way to illustrate this is to recount what the astronauts needed to do on their trip to the moon. So the moon is a fairly desolate-looking place, barely any atmosphere to speak of, in contrast to our wonderful planet. And so to survive, the astronauts had to wear big spacesuits, bringing their life support with them. Now, for you children in the audience, I want to make clear this is a Carlsberg beer ad, and they did not have beer on the moon. Though, of course, the Carlsberg company would like to tell you that it is indispensable for some humans. However, what they did have within their spacesuit was abundant oxygen. And we, as egocentric humans are so concerned with oxygen for our vitality that when we reflect on what's possible for life elsewhere, we often look for oxygen, which makes sense because the Earth has it in great abundance. And so you can see this reflected in the brightness temperature, which is a measurement of the irradiance coming from the IR region. And there are absorption peaks that are diagnostic of particular gases in the atmosphere or particular vapors. And so here you see a peak for CO2, here for ozone, and here for water. And of course, the only planet comparing Venus, Earth, and Mars that has a signature of oxygen and water is the Earth. And thus, a lot of research in planetary science today, where we're looking for signs of life on exoplanets, is very much informed by this, very much looks to see, oh, is there water? Is there ozone? Now, this is all well and good, and of course it's a sign of life on this planet. Yet had we been looking for oxygen for the first billion years of life's history on this planet, we would have missed it. So it's not the only criterion we need to keep in mind. And this is why. So the age of the Earth dates to roughly 4.5 billion years ago. Yet microorganisms evolved on the planet approximately 3.8 billion years. Okay? And what we know from many different types of geochemical records is that it wasn't until 2.4 billion years ago that what is called the Great Oxidation Event happened. That is, we had the first good evidence for the rise of atmospheric oxygen. And for over the first billion years of life's history then, microbes had to contend with either none entirely, or if there was some oxygen present at only trace quantities. Okay, and yet we're human, and so we think a lot about metabolism based on oxygen, and of course here in Woods Hole, we think about going to pie in the sky and consuming that with great abundance and enjoying it, and we have our waste products. And so if we put this on a more cellular, very simple chemical level, and we look inside each one of our cells into our mitochondria, what's going on there is that we're gaining energy from all that sugar in the pie in the sky products by burning that fuel through combusting it with oxygen. Okay, so this is what's called the electron donor, and this is the electron acceptor for aerobic respiration. And I would imagine in this crowd, many of you, if not all, are familiar with the idea that our waste products, CO2 and water, are recycled thanks to the powerful process of oxygenic photosynthesis. Both of these really crucial types of metabolisms, aerobic respiration and oxygenic photosynthesis, were invented, of course, by none other than our friends, the bacteria. And here you can see the chloroplast and the mitochondrion in the lineages from the bacterial domain from which they evolved in a beautiful tree put forth by Carl Woese and elaborated by Norm Pace many years ago that really gives us a picture of the tree of life. So what I would like to emphasize is that while macroscopic animals and plants occupy what conventionally we think about as the great cycles of carbon and oxygen on Earth, really that's obscuring the fact that the bulk of biogeochemical cycles throughout Earth history and even continuing today are driven by microorganisms. And so all of the rest of this tree is microbial, and though some members of this microbial world are very capable and indeed invented aerobic respiration and oxygenic photosynthesis, many of these organisms do things that are completely different. And in my opinion, every bit as exciting and even more powerful in some ways. So here's the key to understanding what goes on, all right? I'm going to try to very simply explain the concept of oxidative phosphorylation, which was figured out by Peter Mitchell in the 1960s and for which he won the Nobel Prize in 1978. So the way it works at the cellular level, let's take this green cell on the left and this purple one on the right, is that there are two substrates for metabolism from which energy can be gained. And if you know a little bit about these substrates, specifically their chemical potentials, and you can see what the difference between these potentials is, you can predict from, first pass whether or not a metabolism could be fueled by a creative combination of burning one through the help from the other. So as I said earlier, and now I'm writing in generic terms, what we have is an electron donor, a reductant in this conception, interacting with an electron acceptor, B oxidant, and then being converted to the oxidized and the reduced species. So this organism goes in this direction and the other reverses it, and you can imagine cycling between any number of elements, sulfur, nitrogen, iron, manganese. It's really breathtaking. So here we have oxygen going to water, the familiar example. I'm going to show you in just a moment a less familiar example of carbon dioxide serving as the electron acceptor and being converted to methane, a potent greenhouse gas. And I'll also give you another example, this time through the assistance of light, of a photosynthetic process done completely in the absence of oxygen, where ferric iron serves as an electron donor for photosynthesis and turns into ferric iron that becomes rust. So how all of these work at the end of the day really comes down to coupling these oxidation reduction reactions to ion transport. And this itself is really quite versatile. So cells have evolved ways of coupling electron transfer through the membrane to either the extrusion of protons, or in some cases sodium, in other cases both. Oftentimes this is done totally in the dark, and sometimes this is aided through an impulse of light energy. Irregardless, what winds up happening is that there is a gradient established around the membrane. It really pumps it up like a battery, allowing it to do work. And so thanks to this charge gradient, in the process of oxidative phosphorylation, an enzyme in the membrane allows work to be done through capturing the release of these ions downhill through this gradient, driving the phosphorylation of ADP by inorganic phosphate to make ATP. And it's ATP that is the key energy carrier for all cells. All right, so let's take a look at one of my favorite examples of this. It's very dramatic. This is the process of methanogenesis, the creation of methane. So this is a process that is driven by methanogenic archaea on this part of the tree of life. And methane itself was discovered by a pioneering scientist, Alessandro Volta, in northern Italy in Lake Como in 1776, the same year as our Declaration of Independence. So what I hope you can see is that Volta is here depicted in his little rowboat, poking his stick into the sediments of Lake Como. And off in this corner of the image, what is depicted is a glass jar in which he has gathered the bubbles coming up from the sediment, and to which he's going to apply a flame. This is a famous experiment. We love to do it here in Woods Hole in Microbial Diversity, and it's called the Volta Flame Experiment. Now, the students in Microbial Diversity now know what's about to happen, but let me show you a very dramatic illustration when it works really, really well. So that's in slow motion, was given to me by Kyle Costa, a former TA in Microbial Diversity, who's now on the faculty of the University of Minnesota. And you can see Kyle in action a couple years ago, just down the Falmouth Bike Path, in a place we lovingly call Cedar Swamp. So kids, you can do this here too. You can do this here too if you get some adults to go with you and supervise. It's really a lot of fun. In fact, it's so much fun. It's what we call some of us call the hazing ritual, others call the bonding ritual of getting the class of microbial diversity to work together very well from the first night of the course. Now, my second general example, before I tell you more about my own research related to this theme, is on a topic that I actually have studied, but I'm not going to belabor the details. What I am going to emphasize is going this other direction, where we take something a little bit exotic, dissolved ferrous iron, as a substrate for photosynthesis. We can create products that change the globe. So in this case, as I said, ferrous iron, through the aid of light, is converted to ferric iron. And ferric iron, at the pH of most natural waters, rapidly speciates and precipitates as a rock, as rust. Okay, and so here you can see the beginning of the experiment. This is dissolved ferrous iron, it's clear. After a few days in the light with some CO2 added and a few other trace elements, the bottle has turned a nice orange rust, and there's absolutely no oxygen in these bottles. Now you may be wondering what this purple bottle in the middle is. That's a culture with the same strain that was used to create the iron rust, only this time it was provided with sulfide as an electron donor, or hydrogen. These organisms are remarkably versatile, and that's one of the things that makes them so much fun. But keeping on the focus of iron, these purple bacteria, as they're so called, thanks to the carotenoid pigments in their membranes, are thought to be responsible for depositing some very, very ancient iron ore formations that we call banded iron formations. And here's an example. This is from Western Australia. This particular formation is about 2.5 billion years old. It's the Hammersley Formation. And as I mentioned, the oxidation step is really critical, because once you get to ferric iron, then the inorganic chemistry takes over, it precipitates as an iron oxyhydroxide, and then goes down what we call the diagenetic pathway to form sedimentary deposits that accrete over time, and these become known as banded iron formations. Now, the genesis of banded iron formations has been a topic that's really interested geologists for many years, because for a long time it was thought that when you looked at their abundance over time, that the rise you see here around the Great Oxidation effect, the Great Oxidation event, reflected exactly that, the rise of oxygen. And the interpretation conventionally was that as cyanobacteria started evolving oxygen, they invented oxygenic photosynthesis. Oxygen then chemically reacted with the ferrous iron in the seawater and titrated all of these massive iron ore deposits. But then came a conundrum, because of course there were deposits that were discovered in remote parts of the world that were 3.6 billion years old. And there's no evidence available in the rock record to suggest that any oxygen was around at that point. And so today, the best available theory, or hypothesis, I think is even better to say, that explains their genesis invokes the action of these metabolically very versatile anoxygenic purple phototrophs. Okay, so they're very important. They helped create some of our iron ore deposits, but they're also present in Woods Hole. And you can see them if you go to the Sippewissett. So this is a photo of my son, who's turning 11 in a couple of weeks. And so this is yet more evidence that I've been coming to Woods Hole for quite some time. And here he is, he was almost 1, playing in the purple sand, because it's cool. I mean, who wouldn't like purple sand? And this purple color, of course, is coming from these purple phototrophs, only here they're utilizing sulfide, because it's a marine environment, which they're getting from sulfate-reducing bacteria deeper down in the sediments, providing them with a reductant to power their metabolism. So anaerobes are all around us. I've just shown you a few dramatic examples, but what I want to emphasize now and go into some detail is where you find them in places that you might not think they should be. And the moral of the story is that you just need to scratch the surface, and there they will be. Because when we're talking about microbes, it's the microbial scale and the microenvironment that matters. It's not what matters from our eyes, it's what matters from their cells' perspective. Okay, so if you were to scratch rodents' teeth or any of your own teeth, in fact, this was beautiful work done by Jessica Mark Welsh here at the MBL, what you'll find is that there's this lovely structured order to what we call biofilm communities, collections of many different species oftentimes attached to a surface and associating with each other. And within these communities are organisms that run the gamut from strict anaerobes to those that can vacillate between anaerobic metabolisms and oxygen to some that may well be obligately dependent upon molecular oxygen. And the same can be said for our guts. This is a really growing area of biomedicine where more and more we're appreciating the importance of a wide diversity of microorganisms within us and on us and how they contribute to our health. But today I want to start with a more simple example of a context and a scientific story of how we came to understand life a little bit beneath the surface. So last year, I took my son into the laboratory. This is just across the street on the second floor of LURB. And I taught him how to isolate what we called aerobic bacteria. It's aerobic, right? It's out in the air. You know, we're not using any sophisticated chambers to exclude oxygen. These are bacteria that double very rapidly, once every 30 minutes. These particular bacteria happen to be super cool because they were red, and so that made it extra fun to work with. But it was a great project for him to learn how to streak single colonies on a plate, which is the way microbiologists historically have done a lot of work to isolate genetic variants, to study isogenic colonies. So here you see, I was teaching him, this is my plate, here are a few of my single colonies, here are his plates, he did, not surprisingly, better than his mother. And here he was doing some microbial art, which is also fun. So I wonder, if you think about this, and the scale here, I should say, this colony is approximately 1 millimeter in diameter. How many cells do you think are in that colony? And I can tell you each cell is about 1 micron in length. Any guesses? Okay, 1 million. I heard of 1 million. Anyone want to raise that? All right, the answer, I mean, this is approximate. It's on the order of millions, tens to 100 million. That's a lot of cells. It's very impressive. But for me, I have a hard time really envisioning what that means unless I look at it. And so here, looking at now just the surface, this reminds me of the lunar landscape. Actually, that's why I started with that lunar landscape. It was inspired by this particular photograph. What you see are some of these hundreds of millions of bacteria just on the surface. And what's very impressive is when you look now at the edge of this colony going down, you begin to have an intuition for just how challenging it must be for these poor cells here in the center. Right? I mean, what are they doing? Do you think that they actually are seeing any oxygen? I don't. And in fact, it's not just a matter of belief, it's a matter of measurement, because we and many others have taken microelectrodes and have been able to measure precisely what the oxygen gradients are. And it turns out that if you go just a few 20 or 30 microns down from the surface, oxygen is absent, is indetectable. Okay, so this was a colony on a plate, but I think the important point that I want to emphasize before I now really embellish this particular scientific story is that biofilms, these aggregates, sometimes of a single species, sometimes a multicellular community of different species, are found everywhere. They are vitally important across so many different facets of our life. So for instance, they will colonize in the roots of every plant and tree. They're very important for the growth of many different types of plants by helping bring nutrients to plant roots. You find them attached to sediments in aquifers, and their ability to interact with some of these sediments can impact the mobility of things like arsenic into potable water supplies. You find them attached to marine electrodes here on an anode that has been deployed off of the Monterey Bay coast in California. And importantly, you also find them within many chronic infections, including the lungs of individuals living with CF. So biofilms are ubiquitous, and because of this, trying to understand how they generate energy in the absence of oxygen is a really important problem. It's of fundamental interest, I think, just for the very simple fact that this is one heck of a cell biological challenge, but it also, the answer to this question has many potentially practical consequences. Okay. So that's what I now want to tell you about, and this is a subject we've been working on in my lab for quite some time. How do bacterial biofilm cells generate energy when oxygen isn't there? Okay, and so in this diagram, you're going to see this now coming back many times throughout the talk. This blue layer represents the part of the biofilm where oxygen is available to the cells, and underneath, this is an anoxic zone. So that's the question. How did these cells survive? And I can tell you they're not dead, okay? They're metabolically active. They're just growing much more slowly than those here that have access to oxygen. So this is a story that began quite some time ago, really when I began my lab at Caltech, and it came from reading a lot of papers with my first student, Mario Hernandez, who was a student of environmental science. And what Mario and I were able to come up with at the time was a hypothesis that something that I'm going to explain to you in just a minute what this means, extracellular electron shuttles, called EESs, that these things may be a very important and potentially broad survival strategy for cells that are oxidant limited. So what is an EDS and how did we get this idea? Okay, so here you see the gist of it. For a cell in the middle that cannot access oxygen, if there was something else in the environment, either organic or inorganic, but something that it did have access to, that it could transfer electrons to, then that reduced molecule could diffuse or through some other mechanism reach an oxidant far away, be it molecular oxygen or an electrode or a mineral of the right redox potential, that it could become reoxidized and then be available again to the cell. And so with this model, what it implies is that you only need a very small amount of this electron shuttle in order to catalyze potentially a lot of metabolic activity on the part of this cell and ultimately a lot of reduction of whatever is the ultimate oxidant at a distance. Okay. So back in the 80s and 90s, environmental scientists were really interested in catalyzing the oxidation of benzene in groundwater aquifers coupled to the reduction of iron. And something that they sort of serendipitously stumbled upon, this was work from Derek Lovely and other environmental microbiologists, Ken Nielsen as well, was the observation that a very ubiquitous class of dissolved organic compounds called humic substances, which are represented here. There's no one structure for humic substances. What's important is that they have moieties called quinones that are capable of receiving 2 electrons and 2 protons from bacteria and then transferring those to an iron mineral. Okay, so these guys observed that when they added humic substances to groundwaters, it really boosted iron reduction coupled to benzene oxidation. And this was very useful in contexts where they were trying to bioremediate toxic organic pollutants in groundwaters. Where we came in was to notice that a standard surrogate, something called anthraquinone disulfonate, used to model humic substances, here I'm showing you its structure, looked very, very similar to things that a totally different community, the medical community, thought of as redox-active antibiotics. And so some of these things you may have heard of, tetracycline, piocyanin, doxorubicin, an important anti-cancer drug. And there are literally thousands of more examples of anthraquinones, naphthoquinones, phenazines that fit this bill. And so Mario and I surmised that maybe, possibly, some naturally produced so-called antibiotics might serve as extracellular electron shuttles. And this wasn't a totally crazy idea because it's been known to environmental microbiologists for a very long time that one of the glories of doing microbiology is that you can isolate bacteria that make beautiful color pigments. And it's not just that they make colorful pigments, but it's that these pigments change color. So sadly, this is just a single snapshot in time of this Munch scream, which I don't think should be viewed as the Munch scream. I think it should be viewed as a celebration of how wonderful, microbial metabolism can be. But if you had this in a movie, what you would see is that some of these diffusible pigments actually change color over time, and that's really the secret. And so to test this idea that, any of these biofilms here making these diffusible pigments might be surviving in part thanks to being able to utilize these lovely redox-active tunable chromophores, we needed to pick a tractable model system. And so we turned to an organism that's notorious because of its importance in many different chronic infections, but yet in nature is just a common soil bacterium trying to make its way. And its name is Pseudomonas aeruginosa. And the thing that I love about this organism, I love many things about this organism, but the thing I love the most is that it makes a class of compounds called phenazines. And this is the core structure of a phenazine. I love chemistry, and I love microbes because their chemistry is so versatile. And one of the things about phenazines is that they come in all these different forms. And I'm just showing you 4 examples here, and you'll see the detailed structures in just a minute. But the point is that depending upon the moieties you have at the X and Y position, that will tune the property of the molecule. So not only will it change its visible spectrum, but it will affect its redox potential as well as its hydrophobicity. And these molecules are actually environmentally significant. They've been measured within infections from 10 to 100 micromolar concentrations. There's evidence that they're made on marine anodes deployed, again, as I said, out in Monterey Bay. And there's been a lot of excellent studies done by environmental microbiologists demonstrating that these compounds are really significant, especially in arid soils. Okay. So a lot has been known about phenazines. And by and large, most of the literature, and there's some important exceptions, but most of the literature has emphasized that they're virulence factors because they react with oxygen. And that's true. When they react with oxygen, as you can see here, one of these phenazines, which is called piocyanin, in its reduced state, it's colorless. And so you see the background color of the medium. In this case, it's yellow. But the minute you introduce the oxygen, it reacts chemically, generating superoxide, and it becomes oxidized. So the medium turns this lovely aquamarine shade, combining the blue and the yellow. And then as these cultures are allowed to sit, the bacteria take the extracellular electron shuttle back up and intracellularly reduce it. And they have machinery that imports it and exports it, and they manage it because these are so highly reactive that they wouldn't be able to survive if they hadn't evolved exquisitely specific mechanisms to keep track of them inside the cell. But it's this extracellular reaction that historically was the focus of a lot of attention, and for good reason. As many of you in the audience appreciate, species are very damaging to lipids, to DNA, to protein. They're harmful. And it's been demonstrated that phenazines are virulence factors in a variety of animal models. All of that is true. But it's not the full story, because if you didn't introduce oxygen, and many of these phenazines are made in the absence of oxygen. Oxygen is not required for their biosynthesis, with one exception. Reactive oxygen species wouldn't be generated. And so that begged the question, might there be other functions for these molecules in the absence of oxygen? So my lab and others have been looking at this now for nearly 2 decades, and I'm going to summarize a lot of work done by people in my lab over many years, including someone in the audience. And what I'll just tell you is, the bird's eye view of this. What we know is that not only can these phenazines, when they leave the cell, react with a variety of oxidants, including oxygen, and generate reactive oxygen species, and that causes toxicity. But if the oxidant happens to be either manganese or ferric iron, it can mobilize metals. When they come inside the cell, they react with specific transcription factors that elicit a program of gene expression that is involved in their cycling and processing. And finally, the part that I'm going to emphasize today is how it is when they come back into the cell, they enable ATP generation, anaerobic survival. So for the remainder of my time, I'm going to tell you about work we've done to figure out how it is that these beautiful pigments help these cells stay alive. So I did fall in love with genetics, and that actually happened thanks to being a student in the microbial diversity course. I found out about transposons from Abigail Salyers, who was the co-director of the course in 1995 when I took the class, and I became hell-bent on learning genetics from that point going forward. So we do a lot of genetics in my lab now, and this was one genetic experiment that was very revealing, and this was performed by Lars Dietrich, a former postdoc. What Lars did was make a mutant, here on the left, that was unable to produce any phenazines. He deleted all of the genes involved in its biosynthesis. Here on the right, he generated a mutant that overproduced these molecules. And here in the middle, we have the wild type. So if you squirt a small volume of these on a plate and let it develop over the course of a week, here's what you see. So this is on the first day. They all look pretty much the same. But after a week, there's a tremendous amount of morphological variation. Whereas the culture that overproduces these phenazines on the right is nice and tight and smooth and thick, that on the left that can't make them is really occupying a very large surface area and building up these incredible wrinkles. And so Lars quantified these wrinkle diameters and stuck microelectrodes into them to measure where, within these different parts of these colonies, oxygen was present. And what we wound up finding was that the phenazine mutant rearranged so that everywhere it had access to oxygen. It either took the Los Angeles strategy of spreading out or the New York strategy of making skyscrapers. But either way you like it, those cells had access to oxygen. Whereas here, the mutant that overproduced the phenazine, they were able to occupy a much larger volume, including volume that was entirely anaerobic. So how does that work? Well, to figure that out quantitatively, one needs to move into a different system. It's challenging to do this in the biofilm context. So imagine, if you will, starting a liquid culture by taking the conditions in the core of this biofilm: high cell density, no oxygen, and concentrations of phenazine that we know to be relevant. So Yuen Wang and Suzanne Kern in my lab took this approach. Here you see an electrochemical cell that they made, where instead of oxygen, we gave the cells an electrode poised at a potential that was high enough to reoxidize the phenazines present in the system. And we added a concentration of phenazines that was not only relevant to these biofilms, but also happens to be relevant to concentrations that we see in clinical samples. And we used a mutant that didn't make any phenazine, so we could really precisely control the amount present in the system. And then all we did was ask, okay, if we give them sugar, if we give them glucose, how well do they survive when we turn on the electrode and we give them phenazine? And it turns out they survive really well. So this is a log scale. Here you see just linear scale time in days on the horizontal axis. And in the presence of the electrode turned on, the phenazine, viability is terrific. Whereas if we either turn off the electrode or we take out the phenazine, we see appreciable death, over 2 logs of death. And so when we did this experiment, not only with piocyanum, but with any of the native phenazines that Pseudomonas makes, we always saw the same thing: survival in the absence of oxygen. So how did that work? Well, I have a very talented former grad student, Nate Glazer, who figured this out. I'm just going to summarize his findings rather than show the primary data, because I want to get to the new exciting data to finish my talk. So what Nate was able to dissect using a combination of genetic and metabolite analyses was that compared to what sustained the cells in the aerobic part of the biofilm, that is conventional aerobic respiration. Now I'm showing you in greater detail than I did at the beginning. The passage of electrons through the electron transport chain, generating that battery, as I said, that can be harnessed to drive ATP formation. That rather than taking this approach, the cells in the anoxic core, and certainly the cells in our anoxic chamber, were doing something I like to call facilitated fermentation, or to be a little bit more technical, facilitated substrate-level phosphorylation. That's what fermentation is, and fermentation is really cool, as some people appreciate here. So not only because it makes beer, but because it makes ATP through really, really cool ways, all right? And so in this case, what's going on is that there's machinery that cycles these phenazines, And the phenazines inside the cell are able to preserve redox balance with the key reductant in the cell, NADH and NAD, this flavin vacillating between the reduced and oxidized state. And in order to, in fermentation, achieve substrate-level phosphorylation, one needs to be able typically to oxidize a carbon substrate and pass those electrons on to something else, usually the NAD plus flavin cofactor. And so what phenazine cycling does is enable this flavin cofactor to be present at high enough concentrations that this metabolic pathway can feed forward, producing ATP inside the cell, which then can be hydrolyzed by the ATP synthase, establishing that gradient. That's how fermenters survive. They have a gradient too, only they don't need it for ATP synthesis. They use it for other things like motility and ion transport and transport of many, many different important chemicals. All right, so that's all well and good. This was something we worked out within these nice chambers that mimicked what was going on in the biofilm, but the recycling is so critical to the story, and the whole motivation for the project came from trying to understand how it works in a biofilm, that we really felt compelled to look at this very basic question, which was central to our model. And that is, if cycling is necessary for these phenazines to sustain metabolism, how do we avoid, or how do these cells rather, avoid the problem of loss due to diffusion? Why isn't this just a loser strategy? Why don't these molecules just diffuse away into the environment? How do they retain them? And furthermore, if they manage to retain them, is diffusion the way that they're going to get around through the biofilm? Is that going to be fast enough to sustain the metabolic rates needed to keep these cells alive? So to get at that, we proceeded systematically to answer two questions. One, are phenazines retained in the biofilm, and if so, how? Two, how do phenazines transfer electrons extracellularly from anoxic to oxic regions of the biofilm? And I'll tell you now, I think I have a pretty solid answer to number one, and we're working to get a really solid answer to number two, but I'm going to show you our best ideas at present. So a big clue that helped tip us off into a possibly satisfying explanation in this context was the observation that not only do phenazines enable biofilm development, this just recapitulates now at a different scale what I showed you before. Here's the wild type, it makes these nice mushrooms. This is a different way of growing biofilms from what I showed you. Here they're attaching to a glass surface and nutrients are washing over them, and you're looking at them with confocal scanning laser microscopy. In the absence of phenazines, they don't make these mushroom-like structures. Well, you add back physiologically relevant concentrations and it recovers the ability. Okay, so that's observation number one. At different scales, phenazines facilitate biofilm development. Critically, however, observation number two is one that had been known for almost 2 decades and came from discoveries by other labs, and that was the reporting that in many different bacterial biofilms, there's copious amounts of extracellular DNA. And interestingly, in this organism, Pseudomonas aeruginosa, it's been estimated that of the biofilm extracellular matrix material, DNA comprises as much as 50%, which is a whopping amount if you think about it. So that really grabbed our attention because, like I told you, phenazines are toxic. They can kill cells, and we wondered, might they, under initial stages of biofilm development, be toxic even to a certain subpopulation of the producing culture? And so Lucas Marias and my lab took on this problem. Here, what you see on the left is just an image of a developing microcolony of a biofilm. In green are live cells, and pink are cells that are dead. And when he quantified earlier, even in the steps toward biofilm development, the death due to 2 different mechanisms that I won't get into, the fraction of cells in the population that were dead in the wild type compared to the fraction of cells that were dead in the phenazine mutant, what he saw was that the proportion of dead cells was significantly greater in the wild type, statistically. And this was attributable to phenazines compared to the phenazine mutant here. All right. Now, as I mentioned, there's this continuum. So when biofilms often form, they're starting to form, and there's plenty of oxygen around, certainly in these flow cells, or certainly when they land on an agar plate in an aerobic environment, they're starting to develop and they're utilizing oxygen. This can lead to lysis, and it's been shown by other investigators that that DNA then can become a scaffold upon which these cells can climb and start to nucleate biofilm formation structurally. Okay, but as this is happening, they're consuming the oxygen, it's becoming hypoxic. And what I had been telling you about earlier was the idea that after a certain critical point, all the oxygen is being consumed by these cells in the outer part of the biofilm at a rate that outpaces its diffusion inside. So what we then asked was, might there be a function for all of this extracellular DNA in retaining phenazines and potentially even facilitating electron transfer for cells in the anoxic interior? So Scott Saunders, a current grad student in my lab, bravely took this on, and he decided to address the first question, which is retention. And so he went back to that simple biofilm model that I showed you earlier, where we grew agar-based biofilms on the surface of an agar plate, and he separated the cells from the agar below by a membrane with a pore size through which the bacteria could not penetrate. And so this allowed him to separate the biofilm from the agar and simply ask, what's the phenazine concentration in the biofilm mass versus in the agar? And what he saw was really fascinating. Okay, so here are 3 of the primary phenazines made by Pseudomonas aeruginosa. This one on the left, phenazine carboxylic acid, is negatively charged, and it partitions roughly equally between the biofilm and the agar. So its retention ratio is roughly 1. Okay? Whereas phenazine carboxamide and piocyanin, both of which are neutral at the pH of relevance to this system, are far more concentrated in the biofilm than they are in the agar. And in fact, piocyanin is concentrated almost up to 50 times as greatly within the biofilm matrix as it is found outside diffusing into the agar. So in vitro, one can do a simple experiment, and that is take pure DNA and mix it with phenazines and ask what the binding constant is. And so using isothermal calorimetry, we found that whereas there was no detectable binding for the negatively charged PCA to negatively charged DNA, as would be expected, we did find significant binding for both PCN and piocyanin, and the greatest affinity for piocyanin, which correlates with these retention ratios we observed. So here's where it started to get really exciting, which was to say, okay, that makes sense. And in fact, these molecules have been described as DNA intercalators for decades. So this wasn't that surprising. But then it got exciting because not only can these molecules intercalate into DNA, but then you start to think, well, is it possible that electrons can transfer through the DNA to these molecules? And might that facilitate electron transfer in the biofilm? So the reason that we thought this was because I happened to be fortunate to work at Caltech, where I have Jackie Barden as my colleague and friend. And Jackie is someone who studied DNA charge transfer for many decades and is very renowned for her work in this area. So with the help of Edmund Say, a postdoc in her laboratory, what Scott was able to do was utilize a special electrode system they had, which they backfilled with mercaptohexanol, such that only electrons passing through the DNA were able to reach intercalated phenazine carboxamide, which when it's oxidized intercalates into the DNA base pair stack. Okay, and so using this system, what we were able to do was do a reductive sweep, and we see current flowing when we get to this critical midpoint potential. Current is flowing here, now reducing the oxidized form to the reduced form. And then when we sweep back, we see this peak corresponding to electrons now giving back from the phenazine molecule to the electrode. And this is what is called a cyclic voltammogram that relates current to the applied potential. Now, what I hope you notice is that there are 2 curves, one in orange and one in blue. And what makes this diagnostic for DNA charge transfer is the fact that you get more current in this orange curve, which corresponds to well-matched DNA, through which Jackie and her coworkers for many years have demonstrated DNA is able to flow. And what they have shown very conclusively is that when you introduce a mismatch into this base paired stack, that attenuates current flow. And so just as we would expect if it really was DNA charge transfer, we saw an attenuation. Now, what got very exciting was that whereas this experiment was performed under anoxic conditions, and really what you're looking at is the amount of current generated by just how many of these phenazine DNA tethered combinations we have packed onto our electrode array. When we did this under oxic conditions, then it became catalytic. Okay, and so the idea is that here now with oxygen around, as soon as the phenazine gets reduced and pops out of the DNA, it gets reoxidized by oxygen, being able to receive more electrons, and accordingly current goes through the roof. All right, and so in orange, this is the same plot I just showed you on the previous slide, only now the scale has changed significantly. And what you see here is a control in black without any of the phenazine added. And so the current really is due to the presence of this phenazine. Even in the presence of mismatched DNA, there's sufficient electron transfer through the DNA in the presence of oxygen that you see an increase in current. But really where it goes to town is with the well-matched DNA in the presence of oxygen, where you get just a dramatic stimulation of current flow. So this was tantalizing, but this is just a very well-controlled in vitro experiment, which begs the hard, big question, what happens in the biofilm? And I warned you, this is something we're working on. This is actually technically very hard, but I'm very excited about it, so I'm going to present to you and end with our most tantalizing recent data. Scott, in collaboration with Lenny Tendner and his postdoc Matt Yates at the Naval Research Lab, who are individuals with great experience in studying microbial fuel cells and electron transfer between microbes and electrodes, they helped us do studies using something called an interdigitated electrode array. It's basically a fancy platform upon which to grow a biofilm, which you can suspend in an anoxic chamber and then control electrochemically. And so the way it works here is that there are two different electrodes, one that we call the source, which is the electron donor, if you will, and here on the right, one that we call the drain, poised at a potential to serve as the electron acceptor. And in between, we grow a biofilm, it blankets, and there are hundreds of these electrodes at a very small scale that are interwoven. So here's a bird's eye view. You see the biofilm coating them. I'm going to zoom in now where you can see actually some of these electrodes in a very tiny part of the IDA. If we take a picture at the surface of the IDA, and then if we just take an image 10 microns up, only 10 microns up from the surface, looking at a biofilm mass, and we stain for extracellular DNA here in blue, cell material here in red, and then we do an overlay. So you see in magenta the bulk of the cell material. The point is that the eDNA is everywhere. It's within the biofilm, it's outside, it's bordering it, it's very, very abundant. And so then comes the big question, can we see whether through these source and drain pairings, we're able to get measurements that help us interpret how electrons are moving through the biofilm? So the first thing is to get a system that you know is really detecting phenazine electron transfer. And so what I'm going to show you now are plots showing the cathodic current, that is electrons flowing to oxidized phenazine off of the source electrode to the drain, okay? And this is the blue line. That's electrons flowing from reduced phenazine to the drain electrode. And we poise the potentials in such a way that when we begin, we're at an oxidizing potential from the source. And the drain is always held at a very high oxidizing potential because it's the electron acceptor. As we move along and we make the potential on the source ever more reducing, we hit the midpoint redox potential for phenazines and they start to flow. And here now when we're at a saturating high reducing potential, we see maximum current. And importantly, what we find in controls are that our phenazine mutant exhibits no redox activity in the system, and the addition of exogenous phenazines restores these electroactive profiles. So this brings us up to the present day, so much so that I even drew it by hand, because we're in the middle right now Scott is back in the lab collecting the data to try to distinguish between 3 different scenarios. And these are not, I should point out, they are not mutually exclusive. And we're very open to what we will find. So one scenario is that within now really the big picture of this is back to the biofilm, going from a reducing to an oxidizing regime, as depicted here. One can envision a pathway that is purely based on physical diffusion. Another pathway might be what we call electron hopping between molecules. And finally, the sort of fantastical scenario of potentially being aided by bounded diffusion or even DNA charge transfer. Due to theory that I don't have time to present tonight, what's exciting is that we should, in principle, be able to distinguish between these mechanisms. And so stay tuned. What I think our current data is currently suggesting is that at high concentrations of phenazines, we're primarily in a diffusive regime, at least in this experimental setup. But at very low concentrations of phenazines, we have some hints that bounded diffusion or possibly DNA charge transfer might be helping out. But again, it's early days, and we'll see what we'll see. But this brings me back to where I started, and really the message that I want to leave you with, which is that in nature, we have microbes found everywhere, in places that might surprise you, and doing things that you might not imagine that they were capable of. And I think that this is an incredibly exciting time to be a microbiologist. There are so many important processes that microbes impact, and so many aspects of how they affect their changes that have tremendous relevance to human health and the environment, that there are abundant opportunities for young people to really make a mark. And so I hope that this lecture has inspired especially some of the young people in the audience into thinking about microbiology and microbial metabolism. I hope it's been entertaining for those of you who know a lot about this topic. I've been trying hard to mention, as I've gone through, all of the names of the people in my lab throughout the years who've contributed. Here on the top, I show the people who have done really foundational work on the project but are no longer in my lab. And here on the bottom is the team that were hot on the chase of the mechanisms whereby extracellular electron transfer actually happens. So I will end with this, and thank you for your attention.
So we have time for a few questions. Please just raise your hand and wait for the microphone. You know.
So you mentioned that that DNA can catalyze charge transfer. So what is the...
Hi, okay.
Sorry. So you mentioned that DNA can catalyze charge transfer. What is the mechanism by which electrons hop through DNA? Is there a particular nucleotide that catalyzes that?
They're thought to traffic through the pi orbital structure in the double-stranded DNA.
All right.
Yeah, and so Jackie Barden's work over decades has spoken to precisely how this happens. Yeah.
Thank you.
Yeah.
Chains of, oh, there it is. How long are these chains of DNA? Are they going from the very center of the biofilm all the way out to the edges, or are they, there are a bunch of little strands in between?
Yeah, that's a great question. So we're actively taking a lot of images to try to answer that. And what I can tell you is it's very heterogeneous. So there are some that are really, really long, like shockingly long, hundreds of microns long, it seems. And then there are some that are shorter. And so that's the reason why I don't anticipate there's any one solution here. I think that it's theoretically conceivable that in parts maybe you have DNA charge transfer, maybe you don't, but that it's not going to be the full story because it's cut up. It's not like continuous, right? But we're trying now to image that. I wish I had access to all the physiology microscopes that just went away because we could have done a really good job. But maybe next summer I'll come back and try that. Yeah.
amazing talk. Thank you so much. Could you give an example in human biology where a biofilm would be of an advantage rather than a disadvantage? People working with CF are familiar, I think, with your work and thoughts about that, but...
Yes. Yeah, that's a great question. Well, my first instinct is to think to our guts, okay? And I believe more and more images are coming out that are starting to give us a picture of what that microbial community looks like. I'll be honest, I haven't seen images yet that would convince me within the gut of precisely what the structure looks like, but that's coming soon, and maybe I'm just ignorant of them, but I can't imagine that you don't find, dense structured communities in the gut, and we know that the gut microbiome is absolutely vital to our health. And so I would think that there's a good case where trying to understand the structure of those communities and their function chemically, this is something of great interest, I think, to some of the students here in this room, at least, would be a great place to begin for that.
Where does the extracellular DNA come from?
It's thought to come from lysis, okay? So, lysis, it means the bursting of a cell from lytic to lyse, to break. And so in Pseudomonas, there have been beautiful movies made of what they call explosive lysis. This is work out of an Australian lab, Cynthia Whiteshirt's lab, and you can do a time-lapse movie and you can watch cells literally pop and explode. And what's thought to be going on is that there's a prophage in the genome that gets triggered that then winds up causing the cell to go into stress and burst. However, it's not the only mechanism, because what we've seen in our time-lapse movies is, in addition to this type of explosive lysis, what we've seen is cells that just kind of contract. And when they do this, DNA just starts to ooze out of them. So their cell membrane loses integrity, and DNA can leak out in that way as well. There may be other ways that I'm not aware of. Phage, if you had in some communities, not only ones that are part of the genome, but parts that are phage that might be coming in from elsewhere. There are a lot of different ways you can get DNA out of these individual cells.
One last question.
Yeah.
Thank you for your talk. Very inspiring. I'm wondering, similarly to the man who was talking about the gut biofilm,
Yeah.
that makes me think that these processes can happen in a variety of environments, such as high acidity, high temperature, low temperature. This might be too soon, but have you tested different parameters to see that there's an optimal amount of charge based on the environmental conditions?
Well, thank you for your question. Something that I want to emphasize that perhaps I wasn't sufficiently clear on is that though we find biofilms everywhere, including in hydrothermal vent systems, where you have, amazing types of organisms doing really great chemistry, what I'm talking about isn't the only way that bacteria can survive. And so there are other strategies they can take. Well, there are a lot of these beautiful redox-active natural products, and there may well be many organisms for whom such a strategy is important. There are other bacteria that do not make these type of redox-active products, and they have other strategies. And that's actually very important for me to emphasize. I would not want people walking out of here thinking the only way biofilms survive is through this type of process, because that would not be correct. So the true answer to your question is that I don't know. I think there are a lot of different strategies, and afterwards I'd be happy to tell you more about what I know of the different ways.
Thank you. And let's thank Dianne one last time. And please join us in the Meg's room.
That was perfect.